here. I know there's a lot of people that are pretty excited about today's presentation from Mingyu. Mingyu Zhang is probably one of the better known PhD students, definitely in my tenure here at Hopkins. I met Mingyu 6 years ago. I've been at Hopkins for about 7 years. He was my second master's student. He's my first PhD student, so this is a very special presentation for me as well. When I first met Mingyu, I often kind of bring this up. He didn't know whether he was going to go into infectious disease epidemiology or cardiovascular disease epidemiology. And it was a new student orientation. And I gave a presentation about cardiovascular epidemiology. And then after that, he followed me to the Welch Center and he said, I'm going to switch to cardiovascular disease epidemiology. And we're very happy that he made that switch. He's found a really special niche within cardiovascular epidemiology. He's focusing on life course approaches to prevention of cardiometabolic diseases from the perspectives of environmental health. I think it's a very interesting area to be in. A lot of papers have come out of his time here. He already published 28 papers, which is quite remarkable. 9 of those first author papers. He also applied for an AHA award and received a 1 percentile score on the AHA award. So that's funded his dissertation work. In addition to his research accomplishments, he's also been outstanding in service and teaching. He's TA'd 17 courses at Hopkins. And for service, he's been a big part of the Epi Ideas group. And also he's been a student rep to the ANC committee and also the faculty committee. In addition to his CV, Mingyu is just an incredible individual. He's a fun person to be around, somebody you want to work with all the time. And I think it's going to serve him well and he's going to be very successful in his future. The next steps for Mingyu are going to Harvard. Like having a child, it's like having a PhD student. So you want to make sure they're going to the right place when they're done with you. And I think he's going to be in good hands at Harvard. So without further ado, I'll let Mingyu give his PhD talk.
Thank you so much for the introduction. That's a really nice introduction. And thank you so much, everyone, for coming to my defense seminar today. And I hope everyone on Zoom can hear me okay. I love walking around the room while I present. So if you don't hear me okay, just let me know. So before I get started, a quick question. By a show of hands, how many of you have ever been to the Walters Art Museum in Mount Vernon, Baltimore? I see a lot of hand raised, almost all of you, right? And how many of you have been there recently, like this year? No one? One, only one person. 2, oh, Sonia, right. Okay, I see a few hand raised on Zoom as well. Well, if you have not been to the Walters Art Museum this year, you must have missed a special exhibit called Majolica Mania. Let me see how I can advance the slides. Right, it tells me the Zoom is being recorded. That's why I was not able to advance it. Called Majolica Mania. This exhibit features earthenware and potteries that people living in Baltimore may have been using in the 1800s. So for example, you can use these 2 plates to serve duck, rabbit, or lobster dishes. And if you want to serve water or wine, you can use that decorated water jug. Well, what's in common about these Majolica pieces is that they were finished with a transparent glaze, shiny transparent glaze. And the glaze makes the colors beneath it so vivid and beautiful. But these pieces are as deadly as beautiful. Part of the reason I quote from the Baltimore Sun article called The Dark Side of Beautiful is that the appealing shine is a result of lead incorporated into the glaze at concentrations of 40 to 60, far higher than the lead concentration that causes brain damage in children. And as a consequence, in the United States and UK, thousands of workers died or were injured in the century when Majolica was used. And victims of lead poisonings were disproportionately women and children. And our city, Baltimore, was a center of Majolica production in the United States. So I'm providing this example to show you that heavy metal exposures have always been with us. And when we think of heavy metals, we always think of lead. But equally toxic are cadmium and mercury. And these 3 heavy metals are among the most toxic chemicals for human health. These metals are ubiquitous in the environment and are common in our daily life. So for example, we can be exposed to lead through air pollution. We can be exposed to lead through air pollution and also through vegetables that uptake lead from the soil. And we can be exposed to cadmium through cigarette smoking and also through leafy vegetables such as spinach or lettuce. And also we can be exposed to mercury through fish consumption, especially the contaminated ones that contain methyl mercury in it. These metals do not have physiological role in our human body and can cause harm at very low levels. They're also very persistent and are not biodegradable in the environment. And that's the reason they stay with us forever. So exposure to these metals have been linked with adverse health outcomes in us human adults, including leading to cardiovascular diseases and also higher blood pressure. But exposure to these metals is even more of a problem in pregnant people. According to Hopkins, metal concentrations are higher in pregnant people compared to non-pregnant counterparts. And about 83 to 98% of pregnant people have detectable levels of lead, mercury, or cadmium. And there's a very high frequency of co-exposure to multiple metals. So here I'm highlighting the word detectable levels because I want to emphasize that these metals can cause harm at very low levels. We call them trace levels. So there's absolutely no safe level for exposure to these metals. And even metals that are barely detectable can cause harm to fetuses. And the reason these metals can cause harm to the fetuses is because they're pregnant. They can release the placenta with fetal tissues. A few years ago, investigators in the Boston birth cohort conducted a study. And what they did is that they tested the metal concentrations in paired maternal and cord red blood cells. And they found a strikingly high correlation coefficient. You can see for mercury is 0.96 and for lead is 0.89. So this study demonstrated that maternal sources of metal can readily cross the placental barrier and accumulate in fetal tissues, leading to fetal exposure. And it also demonstrated that the vast majority of infants in the Boston birth cohort are already exposed to metals even before birth. And the fact that these metals can accumulate in fetal tissues suggests that so we are quite familiar with blood pressure in humans because if we have higher blood pressure, we are more likely to have cardiovascular diseases in the future. But this association starts in childhood, as I showed here. High blood pressure in childhood is a risk factor for cardiovascular diseases in adulthood. A meta-analysis concluded that blood pressure strongly tracks from childhood to adulthood. And a more recent systematic review found that higher blood pressure in childhood is associated with both biomarkers and also clinical outcomes of cardiovascular diseases in adulthood. So this tells us that identifying risk factors, especially blood pressure, can facilitate the prevention of blood pressure and cardiovascular diseases across the lifespan and also across generations. A few studies have examined the association of in utero exposure to lead, mercury, and cadmium with blood pressure in childhood, but the associations are not consistent. And also, most of these studies did not examine the association by child sex, despite sexual dimorphism in blood pressure and risk of cardiovascular diseases. And also, most of these studies only included children who were white. And also these cohorts, a lot of studies in the cohorts, they do not have long-term follow-up. So they only measured blood pressure in early childhood. And we do not know whether the association of those metals with blood pressure can extend beyond early childhood into middle childhood, late childhood, and adolescence. Oh, okay. Is it better now? Let me know, I can go back to the podium. So these are for toxic metals. There's another type of metals Oh, friend told me it's still not clear. And I'll just go back to the better. Now, is it? Is it better now, friend?
Yes, I think it's better.
I'm sorry?
Bring your mouth closer to that mic.
Oh,
When you walk in and you turn to the mic, when you turn away from the mic.
okay. How about this?
Is that better? Yeah.
Okay. So there's another type of metal that are hypothesized to be beneficial for human health. We call them essential trace elements, and manganese and selenium are two of them. So there are many food sources of trace element exposure. So for example, we can have manganese intake through food, such as mussels, hazelnuts, brown rice, pecans, oysters, and pineapple. And for selenium, we can have selenium intake through food sources, such as tuna fish, Brazil nuts, beefsteak, enriched macaroni, rice, and ham. Well, if you are living in Baltimore in the 1800s, these are probably the majolica pieces that you might have used to serve those food sources that I just mentioned. The reason toxic metals can cause harm to the cardiovascular system is they are hypothesized to cause inflammation, oxidative stress, and leading to metabolic dysfunction. And in contrast, manganese and selenium may be protective against protective for cardiovascular health is because they may lead to antioxidation and anti-inflammation, and also they can maintain DNA methylation. And previously in the Boston birth cohort, we showed that higher levels of manganese during pregnancy is protective against preeclampsia, pregnancy cardiovascular complication characterized by high blood pressure and also damages to the kidney and livers. But so far, no study has examined how in utero exposure to selenium and manganese may be associated with child blood pressure. The other thing is metals and trace elements may interact with each other, and they may have synergistic or antagonistic effects on human health. But previous studies that examined how in utero metal exposures is associated with blood pressure all used the quote unquote one metal at a time approach, which means this study analyzed data as if the metals occurred individually. But we know this is not true. As I said a few slides ago, there's a very high frequency of co-exposure to multiple metals. So reading through all previous literature, we're left with insufficient knowledge, but of course also opportunities on understanding what are the interactions of metals and what are the cumulative effects of exposure to multiple metals. We don't know about that. And in fact, in the most recent strategic plan, the National Institute of Environmental Health Sciences, NIEHS, emphasized that study of combined exposures or mixtures most closely replicate the human experience and thus may provide unique insights into environmental health sciences. And following this line of research, accumulating evidence have suggested that metals and trace elements may interact with each other in affecting child neurodevelopmental outcomes. But again, how they affect blood pressure is not clear. So let's take a quick break and let me summarize what I've mentioned so far. I introduced 3 toxic metals, and then I introduced in utero exposure to these metals and elements may be associated with blood pressure in childhood, but the association is not clear. Especially what is not clear is how these metals and trace elements may interact with each other. It's not clear. So this was the aim 1 of my dissertation, in which we assessed the association of in utero co-exposure to heavy metals and trace elements with child blood pressure. For all 3 aims of this dissertation, we used data from the Boston birth cohort, which is an ongoing prospective cohort study that started recruiting mother-child pairs in 1998 at the Boston Medical Center, which is one of the largest safety net hospitals in the Boston area. The Boston birth cohort is one of the largest and longest running birth cohorts, and it has been followed up for more than 20 years. And participants are from a predominantly urban and underrepresented background. So about 60% of the mothers are Black and about 20% are Hispanic or Latino. And we have high quality longitudinal data on child blood pressure because child hypertension is a primary outcome designed as the Boston birth cohort. We measured metals and trace elements in maternal red blood cells collected 24 to 72 hours after delivery. And the advantage of measuring metals and trace elements in red blood cells is because they represent maternal to fetal transmission better than plasma concentrations. And they also do represent in utero exposure because of the long lifespan of red blood cells and also the long half-lives of the heavy metals. And the other thing is they are less affected by hemodilution during pregnancy. So this will give us a more precise estimate of the internal dose of these elements. For blood pressure, we measured child blood pressure between 3 to 18 years of age at all child visits. And we calculated the child age, sex, and height specific blood pressure percentile according to the 2017 American Academy of Pediatrics clinical practice guideline. So this is consistent with previous studies and also with other studies. We did this to make blood pressure comparable across different life stages for the children. And our study population included 1,194 mother-child pairs. We first used linear regression models to examine the association of exposure to each metal and each element with blood pressure. And then we used kernel machine regression, BKMR method, to examine metal exposures. So for those of you who are less familiar with mixture analysis, the BKMR is a novel approach to studying metal mixtures, in which our outcome, child blood pressure, was regressed on the flexible function of the mixture components. And this flexible function allows the potential interactions of the metals and also the potential nonlinear effects of mixture components on our outcome, blood pressure. The BKMR method can provide us several estimates, including the association of each metal and element with blood pressure, the associations of pairs of metals and elements with blood pressure, and also the overall effect of the mixture. So here I'm showing the characteristics of our study population. As I said earlier, all three aims are embedded in the Boston birth cohort. So I'm just going to show this table once. Although the aim 2 and aim 3 have slightly different sample sizes, the distribution of the characteristics are similar enough. So we can see that about 61% of the mothers are Black, and about 20% are Hispanic, and about 53% of the mothers were overweight or obese prior to pregnancy. And for children, about 25% were born preterm, and about 23% were born low birth weight. And the blood pressure measurement was 8 years. So this figure shows the exposure response of each metal and element with child systemic depletion. The other metals and elements are fixed at the 50th percentile. So we can see that there's essentially no association of heavy metals lead, mercury, and cadmium with systolic blood pressure, but there's an inverse association of selenium and manganese with child systolic blood pressure. And then we compared the findings from the linear regression model with the ones from the BKMR model, and we found a similar association. It was estimated in the linear regression model that per doubling of maternal selenium and manganese were associated with a 6.23 and 2.62 percentile lower systolic blood pressure. Well, you may be asking yourself, if the associations are similar between the BKMR model, it's the linear. Why do we bother using BKMR model? Then we just stick with linear regression model. Well, here's why. When we looked at the pairwise association, we found an interaction of cadmium and manganese on child blood pressure. So the left panel shows the association of manganese concentrations with systolic blood pressure by different cadmium levels. So we can see that the inverse association between manganese and systolic blood pressure was stronger at higher cadmium levels. And also on the right panel, we can see the association of cadmium with systolic blood pressure by different manganese concentrations. And we can see that when manganese concentrations are low, there's actually a positive association between cadmium and systolic blood pressure. So as I said earlier, cigarette smoking is a major source of cadmium exposure. So in the next step, we examined whether maternal cigarette smoking during pregnancy modified the association of manganese and child systolic blood pressure. And we did find, in fact, modification here. On the left panel is a figure from the BKMR model. As you can see, the red line shows the mothers who smoked during pregnancy, and the blue line shows the mothers who did not smoke during pregnancy. And it was estimated by, in the linear regression model, that among mothers who smoked during pregnancy, per doubling of manganese was associated with a 10 percentile lower systolic blood pressure. But among mothers who did not smoke during pregnancy, there's no association. So in the first aim, we found that optimizing in utero selenium levels, as well as manganese levels in women who smoked or who had higher cadmium during pregnancy, may protect offspring from developing high blood pressure. And methodologically, we demonstrated that traditional regression models do not account for metal co-exposures, and they actually obscure the true effects of metals on human health outcomes. So had we not used the BKMR method, we may have incorrectly concluded that cadmium is just not associated with blood pressure, and manganese is always inversely associated with blood pressure, regardless of cigarette smoking status, right? So this really emphasizes the need for future studies to focus on how metal and trace element co-exposures affect health, and not just child blood pressure, but also other human health outcomes. I have presented these findings at several occasions, and one question I always get is, what do you mean by optimizing in utero selenium levels and manganese levels? Are you suggesting that we should recommend selenium and manganese levels, or selenium and manganese supplementations for pregnant people? I saw Dr. Miller on Zoom just now, and it just reminded me of this question at one of my presentations. Well, I'm not so sure about it, but apparently some reporters do. Our paper was published in Journal Environmental Health Perspectives last year, and after the paper was published, it received wide media attention, which is good. But one of the reports was like this: eating more manganese is key for building bone density, and well, our study has nothing to do with bone density, but anyway, and fighting inflammation. And here are 11 delicious sources of it. And they said another group that should be particularly aware of manganese consumption, so they used the words consumption, is as a study published in EHP found that children exposed to higher selenium and manganese have lower blood pressure. Well, this is definitely not how we wanted the media to interpret our findings. So come back to the question on whether we should recommend supplementation. I want to first say we need to replicate our findings first. And the second, I also want to emphasize that manganese and selenium can be double-edged swords. So in human adult population, studies have found that higher levels of these two trace elements actually can cause harm to the cardiovascular system. And third, I want to quote Dr. Britton and Dr. Farzan from the USC, who wrote a really nice editorial on our paper. And they said exactly what I wanted to say. As more evidence emerges showing interactions between elements and metals, what do we need to do? What should we do? Should we more seriously consider targeted use of essential elements in populations with high metal exposure? Or should we consider nutritional interventions to mitigate the risk caused by metal exposure? We still do not have a clear understanding of it so far. But hopefully, as the field matures, as we understand the nonlinear relationships, and as we apply more sophisticated methodologies to tease apart these complexities, hopefully one day greater clarity will emerge. And this is also a key goal in the 2020 to 2030. Whether nutrition can mitigate environmental pollutants. So in M1, we observed an association of individual metal and trace element exposures with child blood pressure, right? And now we want to understand the mechanisms underlying such associations. And in the second aim, we focused on core metabolites, and we characterized how in utero metal and trace element exposures are associated with core metabolite profiles. So what are metabolites? Metabolites are intermediate or end products of metabolism. So our body experiences metabolism all the time to keep us alive. function, including when I'm standing and when you're sitting there. This includes the process of our body to convert what we eat and what we drink into energy. So for example, the calories in food and beverages are combined with oxygen to release the energy that our body needs to function. And metabolites are small molecule chemicals that are involved in the metabolism of cells, tissues, biofluids, or organisms. And they maintain cell growth, defense, inhibition, and stimulation. And the reason we focus on metabolites in this study is because they are closer to metabolic changes in phenotypes. Of course, in this case, it's blood pressure. And also, they're also highly modifiable. They can potentially provide us intervention targets if we find any of the metabolites are involved in the etiology of toxic metals or in the etiology of child blood pressure. There are some studies that examined how pregnancy, metal, and trace element exposures affect mother's metabolism during pregnancy. And these metabolites were measured in maternal urine or blood samples. But maternal metabolites do not directly represent fetal exposure. And in contrast, cord blood more directly represent fetal exposure. And their metabolites level may actually reflect how the developing fetus responds to the environment in the womb. So that's the reason we focused on cord metabolites in this analysis. We collected cord blood at birth. I mean, not we, but investigators in the Boston birth cohort did it, and they analyzed the metabolites at the Broad Institute of MIT and Harvard. We used an untargeted approach and identified 397 known metabolites. And after QA/QC, 378 metabolites were included in the analysis. And we annotated those metabolites using the Human Metabolome Database. The analytic sample in AIM-2 included 670 mother-child pairs, and we used a metabolome-wide association approach. So specifically, we used linear regression models to examine the association of exposure to each metal and element with each metabolite. And then we applied bufferoning correction, and we used the false discovery rate as well to correct for multiple comparisons. So after bufferoning correction, we identified 25 unique cord metabolites that were associated with at least one metal or trace element exposures. And I'm going to highlight a few interesting associations that we observed. For lead, we found only one metabolite, piperine, was negatively associated with lead. And this inverse association is actually consistent with a study in rats, not in human, but in rats, that found piperine protected against kidney damages induced by lead exposure. Piperine is a xenobiotic and is a major component of black pepper. And this metabolite has been found in animal studies, again, in animal studies, to reduce insulin resistance, inflammation, and oxidative stress, and also may possess potentially anti-mutagenic effects. This is lead. For cadmium, we found that cotinine and hydroxycotinine, two nicotine metabolites, were positively associated with cadmium. As I said earlier, cigarette smoking is a major source of cadmium exposure. So these associations are not surprising. But what's interesting here is that these associations persisted after we adjusted for cigarette smoking and secondhand cigarette smoking. But these associations could potentially reflect residual confounding because we know that cigarette smoking status is usually subject to social desirability bias, and people tend to underreport their cigarette smoking status. So that's a possibility. And for selenium and manganese, we found that they shared their associations with 6 metabolites, which mostly included amino acids and nucleotides. I'm going to highlight a few interesting findings. One particularly interesting one is they were both positively associated with 8-hydroxydeoxyguanosine. 8-OHDG is a well-established biomarker for oxidative DNA damage caused by excess reactive oxygen species. So this finding is actually opposite to our hypothesis because I mentioned earlier, if you still remember, manganese and selenium are supposed to have antioxidative effects. But here we found a positive association between these two elements with oxidative stress biomarker. So this is interesting. We also found that both elements were negatively associated with guanidoacetic acid and also imidazoles propanate. These two metabolites have been linked to adverse cardiometabolic health in human adults. So this association is consistent and also is biologically possible. And now I want to draw your attention to 6 metabolites I'm highlighting here, which include adenosine, C6-carnitine, C7-carnitine, xanthine, hypoxanthine, and also C36,4-hydroxy-PC. If you could just keep these metabolites in mind for a minute, I will come back to them in just a few slides. Of course, a discussion of all those metabolites and their associations and biological implications is beyond my talk today, the scope of my talk today. I don't have time to cover all of them. But we did publish this paper in Journal Environment International earlier this year. And in the supplemental material, we provided all associations of exposure to and we also comprehensively discussed these associations in the discussion. So we hope that future studies can replicate ours. And one question you might have right now is, just a few slides ago, you were saying that traditional regression models are not good enough and we should use mixture analysis when assessing human outcomes. But why didn't you use mixture analysis in AIM-2? So to answer this question, first, mixture analysis are computationally intensive. So even with the Gaussian predictive process approach to speed up our feeding, it takes days to feed a model with a sample size of 1,000 people for one outcome. And how many outcomes do we have in this analysis in AIM-2? We have 378 outcomes, core metabolites. So in addition to being very computationally intensive, if we are on all outcomes, it will also give us maybe some spurious associations that we have no way to see interactions, or they're just false positive findings. The other important thing is that we demonstrated in AIM-1 just a few slides ago. We showed that if we were interested in the univariate association of, for example, each metal and each metabolite, linear regression and BKMR models gave us similar associations and similar differences in similar estimates. So we do have confidence in the associations reported in AIM-2. And additionally, to assess the potential nonlinear associations, we did use the restricted cubic spline plots to assess the nonlinear associations for each metal and metabolite pairs. But of course, we may have missed the interactions of metals and elements on core metabolome. And I do hope that in the future, methodologies can be developed to address this issue. So, summary. In AIM-2, our findings provide insight on the molecular pathways by which in utero metal and trace elements may alter the metabolic programming and affect child health outcomes. And if confirmed by future studies, our findings can be used to inform potential therapeutic targets for children exposed to heavy metals. For example, the inverse association between lead and heparin is quite intriguing. It is consistent with findings in the animal studies, but again, it is replicated by other human studies to see whether is there a possibility that heparin might actually reduce the internal dose of lead in human body. And also, future studies should also examine how mixtures affect metabolism. During revision, we received a comment from reviewer 2, and the comment was, in general, the data analysis is not deep enough. What is the association between metabolites and newborn outcomes? By doing such analysis, maybe we can find out the relations of metal exposure, metabolites, and outcomes. Well, because as I mentioned, core metabolites in Boston birth cohort were collected after delivery. So their levels may be affected by birth outcomes. So we felt that the association of metabolites with birth outcomes is not something we should look at due to the ambiguity. But we do totally agree that the reviewer's comment makes a lot of sense, because just the associations of metals and metabolites alone do not have too much biological meaning. They're not consequential. They're not solution oriented. what we really should do is to link those metabolites with child cardiometabolic outcomes. So of course, in this study, we examined the association of cord metabolites and blood pressure in food. This is what we did in AIM-3, the third aim of my PhD dissertation. In human adults and also in children, metabolites and their associated pathways have been associated with the onset and also prognosis, blood pressure and hypertension and cardiovascular diseases. And in a recent scientific statement, the American Heart Association also calls for need to use metabolomics to better understand cardiac metabolism and cardiovascular diseases. But so far, very few birth cohorts have data on both metabolites and also blood pressure, especially repeated measures. So far, no study has examined this for metabolites with blood pressure in childhood and adolescence, and also with elevated blood pressure. As I mentioned in the previous aim, cord blood metabolites reflect responses of the developing fetus to the environment. So here we really have a missed opportunity to understand what are the developmental origins of high blood pressure and how environmental exposures may affect blood pressure. And also these associations might also provide us with potentially modifiable intervention targets for the prevention of high blood pressure in childhood. So in the third and last aim, our study population was 902 mother-child diets. And similar to aim 2, we used a metabolome-wide association approach. Specifically, we used mixed effect linear and Poisson regression models, and also we applied the Bufferoni and FDR correction to correct for multiple comparisons. In this figure, I'm showing you the 16 cord metabolites that were associated with diastolic blood pressure after Bufferoni correction. These metabolites included classes such as nucleotides, amino acids, isocarnitins, etc., as you can see in the legend. Well, now you can see some familiar metabolites, the 6 metabolites that I told you to keep in mind a few slides ago, which includes xanthosine, hypoxanthine, C36,4-hydroxy-PC, C6, C7 carnitins, and adenosine. So these 6 metabolites, very interestingly, they are associated with both in utero trace element exposures and also diastolic blood pressure. As for these 2 outcomes, systolic blood pressure and elevated blood pressure, after Bufferoni correction, actually also after FDR correction as well, no metabolites were significantly associated with this outcome. We used both Bufferoni and FDR. Bufferoni is more conservative and FDR is more liberal, but we found a similar association. No metabolites were associated with these 2 outcomes. I do want to mention that in the unadjusted model, we did find some associations, but these associations were eventually went away after we adjusted for confounders. Well, so instead of showing you these 2 empty tables, I'm going to show you the top 10 metabolites associated with each outcome. We can still see a few familiar metabolites. Hydroxycotinine, the nicotine metabolites that was associated with cadmium in AIM-2. Now it is associated with higher systolic blood pressure and also higher risk of elevated blood pressure. C6 and C8 carnitins, 2 isocarnitins, were both associated with higher risk of elevated blood pressure, and also hypoxanthine, which is a nucleotide, was associated with lower systolic blood pressure and also lower risk of elevated blood pressure. So as you might have realized that the metabolites consistently showed stronger associations with diastolic blood pressure than with systolic blood pressure and risk of elevated blood pressure, and we do not quite understand why. We also identified 2 pathways that may be involved in the early life origins of hypertension. The first pathway is related to isocarnitins. So isocarnitins are essential in energy metabolism as a transport form of acid for beta oxidation in the mitochondria. And isocarnitins can be transported to plasma, and their accumulation in plasma can represent dysregulated fatty acid oxidation and contribute to cardiometabolic risk. So these associations do make biological sense. In this study, we found that short and medium chain isocarnitines, for example, C6, 7, 8, and 10 isocarnitines, were all associated with higher blood pressure and risk of elevated blood pressure. Of course, some of the associations reached statistical significance after correction, some did not, and there are also two different magnitudes. But the bottom line is they are all in the same direction, positively. This observation is consistent with adult literature as well. The other pathway is related to purine metabolism. We found multiple metabolites in this pathway, including adenosine, inosine, xanthosine, hypoxanthine, and xanthine. The end product of purine metabolism is uric acid. And uric acid, these metabolites, has been associated with higher risk of hypertension and cardiovascular diseases in adults. And additionally, the entire pathway of uric acid production generates reactive oxygen species and can impair vascular health. So this pathway is also biologically possible. In our analysis, I also want to emphasize that quart urate was positively associated with blood pressure, but this association did not reach statistical significance after correction. So in AIM-3, we found that quart blood metabolites were prospectively associated with blood pressure and risk of elevated blood pressure. And we identified two pathways. One is fatty acid pathway, and one is related to purine metabolism that may be involved in the early life origins of hypertension. It's confirmed by future studies. These signatures may serve as potential predictive markers or intervention targets for child hypertension. Here I show again the complete conceptual framework of this PhD dissertation. So in this first aim, we examined in utero metal and trace elements with blood pressure in childhood. In AIM-2, we examined how those metals are associated with metabolites. And in AIM-3, we examined how those quart metabolites are associated with blood pressure in childhood. So taking one step back and looking at the complete conceptual framework, we can see that quart metabolites may explain the association of in utero exposure on child blood pressure. So we just took one step back, and now I want to take one step forward by saying that the significance of AIM-2 and AIM-3 actually extend beyond the association of in utero metal exposure and blood pressure. Our findings in AIM-2 can help us understand how in utero metal exposures may affect other child health outcomes, not just blood pressure, but also neurodevelopmental outcomes, for example, and obesity. And for AIM-3, understanding how these metabolites are associated with blood pressure in childhood also provide us a broader opportunity to understand how in utero other environmental exposures, not just metals, but PFAS, but air pollution, other environmental pollutants may affect child blood pressure. So findings from these two AIM can contribute to understanding of these associations as well. Our study has a few challenges, of course, but we did find some solutions to address these challenges. So first of all, our study is an observational study, and we cannot eliminate the possibility of residual or unmeasured confounding, right? So what we did is that in all three aims, we adjusted for a comprehensive set of confounders, which include maternal cigarette smoking, maternal fish consumption, and also maternal diet during pregnancy. And second, we measure blood pressure once at each visit. And the key guideline recommends that we measure 3 times and take the average of the second and third reading. So having only one blood pressure measurement may have average a higher blood pressure in general. But the good thing is that such errors were unlikely to be differential by the exposure status. In AIM-1, it's metals and trace elements, and AIM-3 is quart metabolites. So if any, these errors would have biased the results towards the null than away from the null. The third is our study is a longitudinal study, as that has been followed up for more than 20 years. So loss to follow up does exist. And also, because we measured metabolites and metals using biosamples, we only measure them in a subset. So selection bias may also potentially exist. How we addressed loss to follow-ons and selection bias is that for all three aims, we compared the characteristics of children and mother included in the analysis versus those excluded from the analysis. And we used the inverse probability weight method to address the potential bias caused by loss to follow-up and selection bias. And finally, one advantage of our study population is that it's rich in Black and Hispanic mothers and children, but on the other hand, we also do not have a lot of participants who are Asian, who are white, or who are other race ethnicities. So this might limit the generalizability of our study findings. So we would like to replicate our findings in Project Viva, which is also a prospective cohort that started recruiting mother-child pairs in 1999. You can see it's in a similar time framework as the Boston birth cohort. And it's even same geographical region in Boston, Massachusetts. And in Project Viva, about 60% of the participants are white and about 15% are Black. And of course, I cannot do a presentation without acknowledging ECHO. We may also want to use data from the environmental influences on child health outcomes. I guess you should be very familiar with it. It's a consortium of 69 cohorts of more than 50,000 mother-child pairs in the United States, and they represent a wide geographical region. And our preliminary analysis found that a lot of cohorts do have data on metal exposure, metabolism, and child blood pressure. And of course, our study has strengths and innovations. First of all, we had a longitudinal study design that allow us to examine the exposure response functions of metals with blood pressure and also core metabolism with blood pressure. And also we have a racially and ethnically diverse study population. This makes our findings especially relevant to these populations. Actually, they're a very disproportionate burden of environmental exposures, also have a higher risk of cardiovascular diseases, but these populations are historically underrepresented, historically excluded from previous research. And third, we have a large sample size. Two things. First, a large sample size allowed us to examine the potential effect modification by child sex, by maternal race ethnicity, and also by child age. I did not have time to cover all those associations, but I do have a backup slides for that. And we can also refer to a publication for this race, sex, and life stage specific associations. Also, we have a large number of repeated blood pressure measurements from childhood to adolescence. And this also minimizes the possibility of the measurement of associations. And fourth, of course, we use the mixture analysis to analyze metal and trace element co-exposures that revealed some associations that we may have missed. And the last is that the metals are measured in red blood cells. And I explained earlier, they can give us a more precise estimate of the internal dose of these metals and trace elements. Our study has public health impacts. First of all, it advances the NIEHS authority to study the impact of chemical mixtures on human health. And second, it can facilitate a shift from treatment to primordial prevention of cardiovascular diseases at the earliest developmental windows. For the second point, it also advances the American Heart Association's recent goal to shift the focus towards preventing diseases before they. Third, of course, this has to be replicated by future studies, but our study can generate potentially translatable metabolite targeted solutions for reducing metal toxicity in child cardiovascular health. And it's actually not just child cardiovascular health, but other child health outcomes as well. And it also provided insight into the early life origins of hypertension and potentially unusual exposures on child health. And finally, our study can inform policy making regarding the risk assessment of metal exposures and guide targeted policies for metal reduction. Well, for the last point on informing policy making, we are not just talking the talk. We are confident to say this based on our previous experience. So my first ever publication was on the association of prenatal exposure to air pollution with blood pressure. After this paper was published, it received wide media coverage. But more importantly, this work has been cited in multiple guidelines, including the ones from the World Health Organization and the ones from the American Public Health Association. And this research has also supported a few statements or lawsuits for children who are disproportionately affected by environmental exposure, by climate change, and by fossil fuels. So we are confident that the knowledge generated from this PhD dissertation provided evidence on how environmental toxic metals may affect child health and how our environmental regulation policies should not be lifted or rolled back, but only should be more stringent going forward. This is especially important given the rule yesterday. You might have seen the news on the EPA versus West Virginia. 10 years ago, I ended my proposal seminar with a quote from the late Congressman Cummings. What he said was that our children are living messengers for a future we will never see. How are we doing on that? NBC News, 95% of baby foods tested contain toxic metals, lead, mercury, or cadmium. Consumer Reports, there are concerning levels of heavy metals in almost half of the tested juices. CNN News, there are dangerous chemicals found in food wrappers at major fast food restaurants, like the ones wrapping your hamburger may have toxic chemicals in it. And Vox, it's not just Flint. Every major American city has hazardous amount of lead hurting kids. We still have a very long way to go. Well, today I want to end my presentation. Sorry for those who arrived late. The Majolica Mania collection at the Walters Art Museum earlier that were glazed with lead that's make them so shiny. But anyway, let's just think about 200 years later. It's the year 2222. The Walters Art Museum is going to have another exhibit called The Dark Side of Beautiful, early 21st century collection. Instead of these Majolica pieces finished with a shiny, beautifully transparent glaze, what will be in their collection? Well, I have so many people to thank. This is the end of my presentation. But first and foremost, I want to thank my advisor, Dr. Noel Mueller. You guided me through everything I needed to know about research, and we just had, oh, I forgot to advance the pictures. We just had so much fun together, and it's truly an honor to be your first doctoral student, and I just wish to have the same impact on others in the future as you had on me. And I want to thank Dr. Wang. You provided me with unconditional support, guidance, and wisdom in pursuing my research and also my career goals throughout the 5 and 6 years we have worked together. I have told multiple people, just like when I'm stressed out, just even after talking to you, I just feel much better. Yeah, I'm also so grateful to Dr. Buckley, who's here today, and Dr. Goyara, who's on Zoom, who you served on my dissertation committee and provided me so much guidance and throughout my PhD dissertation. And I also want to thank my other faculty members who served as examiners and alternate examiners on my oral exam, Dr. Apple, Dr. Kress, Dr. Dickerson, Dr. Grady, Dr. Matsushita, Dr. Benjamin Nillen, Dr. Shin, Dr. Leda Costa, and Dr. Wills Karp. And I cannot thank enough my mentors and collaborators in the Boston Birth cohort, in ECHO, and also in the Welch Center. I also want to thank the Academic Support Corps, especially Fran, especially Laura, and also especially Matt, who helped me so much throughout the years. I want to thank the doctoral seminar faculty and my amazing master and PhD cohorts. And I want to thank my friends at the Welch Center. And I also want to thank the Happy Ideas group. I just felt so fortunate to have joined the Happy Ideas group. And I also want to thank everyone in Mueller Lab group, especially Maura, especially Tinker, and especially Curtis. And oh, this is like a gallery. I don't have to say anything, but I just want to thank my friends, a lot of them who came today. Some drove all the way down to Hershey this morning, some are on Zoom. It's midnight in Beijing. I just had so much fun, and I could not have done this, survived through COVID without all your support and compassion. And I want to thank my parents. Parents. I have not seen them since 2020, since the pandemic started. So I really miss you, and I hope to see you soon. And my grandparents, and of course I want to thank the support during my PhD. I received these honors and awards, and also I want to thank the American Heart Association for providing me the predoctoral fellowship that supported this work. I only have one more slide. I don't have any words on this slide because my grandma cannot read English. I just want to dedicate this dissertation to grandma who passed away last year. I grew up living with my grandma. I spent almost my entire childhood. She was the one who cared for me, who cooked for me, and who stood up and fought back when other kids bullied me. So she was my world. And I chose this picture because it says in Chinese, 且行且珍惜, cherish what you have at the moment. And since I came to the US 6 years ago, I've only seen her like 5 or 6 times, and each time is only like a couple of days or a couple of hours. But I enjoyed every second I had with my grandma, and I wish she could share this moment with me. But I really hope in the future, whoever reads my dissertation can remember she's such a loving and caring person, and she shaped who I am. And without her, I definitely would not be who I am today. And I just want to thank everyone again for coming to my defense in person and on Zoom. And I would love any questions. We only have 2 minutes left, but I hope I can have 1 or 2 questions. Thank you, everyone.